Good morning, everyone. We are happy to have you here in our advanced webinar on spectral flow cytometry. I'm Nitzan, I'm VP of New Technologies and Implementation at Almog. Next to me is Aviva. She's our flow cytometry service team leader, application specialist, and scientific support, and she's definitely our flow expert. So before we will dive into our talk today, we just want to give you a teaser to how you can create a really nice and informative analysis of phenotyping panel using Cytek technology. You can see here a full map of 40 markers expressed across all populations. It is truly a milestone for the flow world, as it is the first time that the flow cytometer has been able to detect so many fluorochromes in the same tube, and it makes you curious as to what the future will bring. I will start with just the agenda for our talk today. We will discuss the differences between conventional and spectral flow cytometry. We will show you the spectral signature and how it is created. We will explain spectral unmixing and compare it to conventional compensation. We will discuss the use of autofluorescence extraction and its advantages in dim population analysis. In the last part, we will show you the advantages and possibilities of this technology by showing you the Aurora 5 laser system. And last but not least, you will see the new and updated OMIP 69 with 40-color immune panel that was recently published in Cytometry 2020. We will start by looking at the diagram of a conventional flow cytometer. As you all know, we have the sheath fluid going through the laser beam. We have the filters followed by the detectors. And one detector is assigned to one fluorochrome. The main difference in spectral flow is the way that light is being collected. First, let's look at some of the limitations of conventional flow that we overcome with spectral flow. As I showed you, there is one detector for one fluorochrome, meaning that we have to match between dye and filter. There is limited combination of dyes. Highly overlapping dyes cannot be run together in the same tube. We have a lower sensitivity since less photons are collected for each dye. High autofluorescence interrupt, especially with dim signals. And of course, there is the complexity of compensation of large panels, spreading and artifacts. So what are we looking at? Fluorochromes typically have more than one excitational wavelength and emit over a range of wavelengths. So when using the conventional idea of one laser, one detector, we're actually capturing only the peak emission. And it's the same as looking at the tip of the iceberg when there is much more beneath it. Let's take, for example, Fitts emission spectrum and the detector bandwidth. You can see here that the detector collects only the peak emission. It makes us assume that Fitts is green. Anything that is outside of the detector is either being compensated or ignored. So, now let's start talking about spectral flow cytometry. It was the idea of it was first introduced to the flow community in this article in 2013, and this article discusses the way that light is being collected across a detector array and not just through one detector. If we're looking again at the FIT's emission spectrometer, FIT's example, you can see that looking at the entire spectrum, it can be the emitted light is collected in each and every detector. And if we're looking at the full spectrum, we can now understand that FIT's full emission goes from 490 to around 650. So it goes from blue, green to yellow, orange, and it's not just green. So let's dig in deeper to full spectrum cytometry. We have the optical design, which is highly sensitive. The laser is spatially separated. We have, for each laser, APD detector modules that consist of several detectors. The APDs are highly sensitive. We'll discuss in the next few slides. The entire emission spectrum is captured across the different modules to create the spectral signature for each dye or prohorm or cell you will see later. And we have the spectral unmixing algorithm that replaces the conventional compensations that you probably know. So a bit about the APDs. The Aurora and Northern Light use APDs instead of PMTs for all instruments. The sensitivity of a detector is being measured by quantum efficiency. It is the ability of a detector to convert photons into electrons and finally to a signal. So you can see that APDs have much higher quantum efficiency than PMTs. And looking at the graph here, the efficiency is calculated by percentage. And you can see that the efficiency of the PMTs dropped down very quickly in about 500 nanometer wavelengths. And if we take, for example, the Alexa 647, you can see that the efficiency is about 5%, meaning that 100 photons will be converted to 5 electrons only. And when using APDs, the efficiency is very much close to 100. So that makes us the sensitivity and resolution in the analysis also. Another important thing is that Cytek is using flat top lasers. This means that the energy is distributed homogeneously. It doesn't matter if you're using low, medium, or high flow rate, you will have the same energy, the same MFI, and the same resolution. In order to show you how the spectral signature is created, let's look at 5 laser Aurora optic design. You can see the range of detectors in each detector module for each laser. The lower the excitation wavelength, we have more detectors in the detector module. So you can see here that for the UV and for the violet laser, we have 16 detectors. We have 14 for the blue, 10 for the yellow-green laser, and 8 for the red laser. Another important thing to mention here that we have 2 side scatter in this configuration. We have side scatter on the blue, and we have violet side scatter for small particles. Let's take a closer look at how the spectral fingerprint is built. You can see the emission signal in each and every detector. In this example, we have 16 detectors, just taking as example the UV laser. And to understand this better, we'll put out just one of those channels, and looking at the top-down view of regular count histogram, you can see that the red area has the most counts, meaning the most dense area of the most dense population, and the blue has the less counts. Okay, so first thing to understand about the signatures, that the spectral signature must be unique in order to distinguish between different dyes. We have here just 4 examples. Here we have the FITs that you can see that the emission spectrum is going through the blue channels, but also through violet channels. We have the P and the APC that emits to violet, blue, yellow, green channels, and of course the APC emits also in the red channels to the red laser. The first most important use of this signature is to evaluate dye compatibility. We have here just two examples. You can see on the left the APC and the Alexa 647 that normally when you're using conventional flow, of course, you consider them the same as they have the same peak emission and you never use them together in the same tube. But when we collect the light across all detectors, the full emission spectrum, you can see that there are quite few differences that can allow us to use them together. On the right, we have the example of FITS and Alexa 488. You can see that the signatures are much more similar, and it's also seen in the overlay histogram. And another important use of the signature is to evaluate spread or a spillover between two dyes. You can see here two colors, PSI5 and APC, that normally you know that you read them on different lasers. The PSI5 you are using on the blue laser and the APC on the red laser. But if you're looking at the entire spectrum, you can see that the PSI5 has a lot of spillover into the APC. This tells us first that if we're using conventional flow cytometer, we still need to run compensation between these two dyes. Although you are using them, you are detecting them on different lasers, still the spillover has to be compensated when using conventional flow. Of course, when we are using, when we're running them on spectral flow cytometer, we have the unmixing algorithm that also resolve this for us. We now hand over to Aviva.
Yes. So, hello everyone. My name is Aviva, and I will talk about some technical and scientific aspects of spectral flow. Now that we have talked about fluorochromes and how we can obtain unique signatures, let's talk about spectral unmixing and how it works for our system. Okay. So here we are again looking at the signatures to think about how the unmixing algorithm works. Here, this signal is a combination of several fluorochromes. And how do we determine how much of each fluorochrome is contributing to this signal here? So very similar to conventional flow cytometry, where single-stained samples are used for compensation. Here in spectral flow cytometry, we use reference controls, which are also single-stained controls, and an unstained sample. We need the single-stained controls in order to collect the signature for each dye. So the spectral unmixing algorithm calculates the contribution of each known fluorophore spectra to the total collected signal in each detector. So when you think about the algorithm, we can think of it as extracting each component until we have nothing left. So here, let's compare between the aspects of compensation and the spectral unmixing. So when we are looking at conventional compensation versus spectral unmixing, it is quite clear that these different algorithms achieve the same thing. It's all about assigning photons to the correct fluorophore. So in compensation, each fluorophore is detected in one detector, and the number of detectors is equal to the number of fluorochromes. Whereas in spectral unmixing, each fluorochrome is detected in all detectors on the instrument, okay? So in conventional compensation, the algorithm mathematically subtracts the spillover, the amount of spillover into primary detectors. In this case here, the algorithm mathematically subtracts the fit spillover into the PE channel. Whereas in unmixing, the algorithm determines which combination of reference controls best fits the signature of the multicolor sample. So here, let's look at the performance of unmixing and compensation of two heavily overlapping dyes. We have here a panel that contains 4 markers, 4 fluorochromes, and we are looking at 2 fluorochromes, the Q.705 and Brilliant Violet 711, that their emission peaks are heavily overlapping. So when we are looking at compensation, we can see that actually the compensation fails, and mostly because you can see that there is much spillover spreading seen, and the positive populations are not well resolved from the negative ones, and the populations are not well clustered. So when we are looking at unmixing and how unmixing deals with these 2 colors, fluorochromes that have almost identical emission peaks, but thanks to the fact that they have different signatures, we can see that the populations are very nicely resolved. And of course, we don't see this heavy spreading of the signals, and negative signals are correctly clustered here. So unmixing has the power to give us high quality data. Okay, so let's summarize here some of the advantages of spectral unmixing. So overall higher resolution is achieved by using unmixing because more data points are collected for the measured particles. Since more photons of light are collected over the full spectrum, it gives tighter clustering of negative and positive populations. We get more colors with less lasers. So as long as the full spectrum signatures are unique, you can overlap, you can use very highly overlapping dyes. Any dye that can be excited by the laser, by the onboard lasers, can be used together, and there is no need for optimized filter sets since signal is collected in all channels. The unmixing also allows us to extract the autofluorescence. The autofluorescence contribution to background noise can be minimized by extracting its signature from the sample. So we can treat the autofluorescence as any other parameter or fluorochrome in our case. So let's look here on autofluorescence of free cell types. We are looking at splenocytes, lung cells, and yeast cells, while splenocytes have very low autofluorescence in all channels, let's say. The lung cells and yeast cells have quite high autofluorescence, especially in the violet laser channels. So, and also notice that the signatures of the autofluorescence are unique. So we can extract these autofluorescent signal by unmixing, same as with fluorescent dyes. Here is a very nice example showing human HeLa cells, which carry an M-cherry reporter gene. We are looking here at the signature of the unstained cells and the signature of the M-cherry. Look at these 2 signatures, they are quite similar. And then looking at the yellow-green channels, their M-cherry signal is collected, you can see that high autofluorescence is seen also in these channels. So when we look at our unstained sample, we really see a quite high signal coming from the autofluorescence of the cells. So without autofluorescence extraction, we cannot gate on our M-cherry positive signal very well because it cannot be distinguished from the negative signal. But after autofluorescence extraction, look how nicely the positive events come up, and now we can gate on our positive population. So I think that this example demonstrates very nicely that this technology is highly sensitive for dim populations. So now that we have talked about fluorochromes, their unique signatures and unmixing, let's talk about the wide range of possibilities using Cytek instruments. So full spectrum analysis enables us to use highly overlapping dyes. This is something that Nitzan has already mentioned in her session, but now I would like you to walk you through the superpower of spectral flow cytometry. So when we are looking at the emission peaks of these two dyes, it is clear that they cannot be analyzed together in the same tube by classical flow cytometer. But when looking at the emission signatures of these two dyes in the eyes of Aurora, very clear differences are seen. Let's overlay these two signatures. And as you can see, there are many differences seen in the ultraviolet, violet, and blue laser channels, but even in the yellow, green, and red laser channels, there are some differences seen. So these differences are exploited in order to combine these two dyes together in the same tube, and even to analyze co-expressed markers. Like shown in this example here, we have CD8 APC and CD57 Alexa Fluor 647, and a very nice resolution of double positive population can be seen here. So even co-expressed markers can be analyzed together than using these heavily overlapping dyes. Okay, so another advantage of this technology and of Cytek instruments is the consistency of the signals across different instruments. So this consistency is of high importance for biotech companies that need standardized data between different instruments installed in different sites. This is possible thanks to the fact that you don't need to adjust the voltages. Okay, so let's look at the signatures. Okay, these are 6 dyes here listed here, and the signatures are shown here for 2 different units. And as you can see, they are completely identical signatures. So this is achieved thanks to the voltage that is controlled by QC beads and optimized Cytek assay settings rather than by the user itself. Another nice thing is the analysis of small particles. Small particles analysis is, of course, achieved thanks to the powerful lasers, powerful violet laser, and thanks to the highly sensitive violet Cytek detector. Like Nitzan already mentioned, the Cytek detector is under the violet laser. So here we are looking at the murine leukemia virus, which express the GFP fusion protein, and the size of the virus is around 120 nanometers. So we can see here how nicely the positive GFP positive virus particles are resolved from the background. We can even go down to 80 nanometers. Here we are looking at 81 nanometers non-fluorescent polystyrene beads. And here we see a very nice resolution of the beads from the background, and this is achieved by scatter signal only, okay? So this is, of course, of, yeah, it is very, very useful also for exosomes and microvesicles that are of high interest today. Another very unique feature that is a real superpower is the ability to subtract the autofluorescence, okay? And this is of high importance when we have dim populations. So high resolution of dim populations is again thanks to the lasers, the powerful lasers, the sensitive detectors, but mainly because the unique superpower to extract the autofluorescence. So actually in conventional flow, what we do usually, we choose brighter colors for dim populations. But this is not something that we can always control. And in this case, really this superpower of autofluorescence subtraction can really help us and to resolve the dim populations. In this example here, messenger RNA of a low expressed gene in lymphoma cell line was labeled with Alexa Fluor 488, and it was run on Aurora instrument. As seen in the spectrum signatures here, the signature of the unstained cells and the signature of the Alexa Fluor 488, they are almost identical. Okay, so they are really heavily overlapping. So on the right here, we can see that when we are trying to resolve the positive signal from a negative one, it is really almost impossible, okay, without autofluorescence extraction. But after autofluorescence extraction, look how nicely now we can get on our positive signal, on our positive events. So this autofluorescence extraction is really a superpower, and it is extremely beneficial when analyzing dim populations. So in spectral flow, there are a wide range of possibilities. With spectral flow, the paradigm has changed. You don't have to think anymore of how many lasers and how many detectors you have. Instead, you should ask yourself how many markers you want to analyze in one tube. So here is an example for 10 violet dyes panel for PBMCs. And these PBMCs are stained with these 10 antibodies that are read on a Cytek northern light instrument that has only one violet laser. And again, we can look at the signals here at the monocytes and look at the TCR gamma delta positive events. And of course, the Tregs and all the signals are very, very nicely resolved here. And this is only with one laser, 10 dyes with one laser. Of course, there is another option of 21 colors panel with just 2 lasers, and it goes up and up. We can today achieve 40 color panels on 5 laser instruments. So you really don't need to worry about the optical configuration of the instrument, but because any dye excited by the laser can be analyzed. Here, let's summarize some of the things that we have talked about. These are actually, we have not talked about the volumetric counting, but we have volumetric analysis of sample in all Cytek instruments. And this is something that gives us absolute concentration of your cells in all readings in all samples, for all samples. The resolution is not impacted by flow rate thanks to the flat bottom beam. This is something that Nitzan mentioned in her talk. One of the easiest things about the Aurora is that you don't usually need to adjust the gains for the detectors. The Cytek assay settings and daily QC automatically set the gains for you. You have an option for plate reader for 96 well plates. It is easily changeable between plates and tubes, and it is done in a matter of seconds. And also washing and mixing between samples are offered. You have automated QC and cleaning steps. There is a very, very useful tool offered by Cytek. It's the Spectrum Viewer. Okay, this is something that is available in Cytek's website. And this Spectrum Viewer shows you the level of similarity between different dyes, and it helps you to decide if you can run them in the same tube. IVD instrument is available today for the Northern Light instruments, okay? And it is really amazing that Cytek instruments are very well established today. They are 3 years in the market, and there are more than 400 devices sold. There are many collaborations with companies like BioLegend and Thermo, which develop new innovative fluorochromes. And Aurora's rapid acceptance in the market, it's a true testament to Cytek's remarkable technology innovation. And of course, Cytek have also established partnerships with software companies like FCS Express from Denovo, Astrolab, Verity, Omic. All these companies are offering softwares, flow analysis softwares for advanced analysis of flow data. For example, here you can see a 40-color panel that is shown in this multi-dimensional analysis platform. It is called TSNE. So, all these softwares also offer a very automatic platform for analysis, and it makes it really easier to gain all this information from these big panels. So now we will briefly look again into Aurora 5 laser system in order to understand its performance characteristics. So here is a nice image of the Aurora instrument, you can see that in spite of all the many features that it offers you and all the possibilities and capabilities, it is a benchtop instrument. It can be placed on any bench in any lab. And of course, the many detectors that are incorporated inside the instrument, these are APDs that are much smaller in size than the regular PNTs. And this is the reason that we can really gain everything in this quite small device. Okay, so here we are looking at the configuration of a 5-laser Aurora. Again, we see our laser, 5 lasers, and we see all the detector modules. We see the ultraviolet, the yellow-green, the red, the blue, and the violet detector modules. And the side scatter detector, which is under the blue laser and also under the violet laser. And this is something that enables us the small particles analysis, as we have mentioned before. Here is a filter configuration. We have overall 64 detectors for the 5-laser Aurora, and each detector has a filter in front of the detector, and the filters have a bandwidth between 15 to 35 nanometers, and it actually captures the emission light between 365 nanometers and until 830 nanometers. Here is a nice example of building a 16-color panel and how it looks in the Spectra viewer in Cytek website. So this is the spectral overlay of all 16 fluorochromes that we have chosen in this spectral viewer. This is one way to look at it, but I think that much more information can be gained from this matrix here that shows us the similarity indexes between 2 dyes, and we will get the overall complexity of the overall panel of all fluorochromes. So the similarity indexes are indicated here, and we can go up until 0.98 for 2 dyes in order to measure them together in the same tube. For an example, APC and Alexa 4647 have a similarity index of 0.96. So this is a very useful tool, and that helps us to combine and choose colors, fluorochromes. So this brings us towards the end of our presentation. I would like to show the new OMIP 069 that was published recently in Cytometry Part A. For those who are not familiar, OMIP is for Optimal Multicolor Immunofluorescent Panel, and this is a special review publication that reports on newly designed and optimized multicolor panels for flow. So Cytek have recently published this review, summarizing 40-color panel for deep immunophenotyping of major cell subsets in human peripheral blood. So let's look here on the panel that was used and that was carefully built. As you can see, we have there are several dyes that are heavily overlapping dyes actually for Brilliant Violet 421 and Super Bright 436, and also FITS and Brilliant Blue 515, and of course APC and Alexa 4647. So this panel was used to stain all these cell populations. I am sorry. And of course, yes, you can see that there are still many channels available here that can be occupied by new fluorochromes that are hopefully under development today. So there is another way to look at the panel. Here are the immunophenotyping markers for all the cell populations that had been chosen. And here we can see the outcome of the analysis. So this is the classical way to analyze this data with density plots. And of course, we get these nice analysis for monocytes, for non-classical, intermediate, and classical monocytes. You can see also the TCR gamma delta positive cells. We have many cell types here: B cells, dendritic cells, and also central memory CD4 positive cells. So everything is really nicely resolved here, and this is a really impressive data, I think. So let's summarize what we have talked today. We talked about the full spectrum analysis and how it helps us to predict dye compatibility. We talked about full spectrum cytometry and what it offers us, the great flexibility, combining dyes that cannot be combined in conventional flow cytometry. We talked about autofluorescence extraction and what it gives us. It gives us much better resolution, especially for dim populations and for cell types that have high autofluorescence. So we get this high sensitivity for dim populations, and we get high resolution for small particles. And all this, we get high quality data with high parameter data that we can analyze on different platforms today. And don't think of how many lasers you have, but how many dyes you can combine in a given configuration. So this is the end, and now you have time for questions. We would be happy to answer any questions you may have. You can type them in the QA section. You can always email us also. We would be happy to answer any questions by email.
There is a question, how many flow homes can be detected with 1 or 2 lasers? So of course, it depends on the configuration, but if we're talking at, for example, one on the blue laser, we have 14 channels. Until now, Cytek has measured a panel of 9 colors with only blue laser. With 2 lasers, they already detected 21 colors in the same.
I think it's even more today. It's 24 color panel.
Yeah.
So, yeah.
Also, it depends on which two lasers. Yeah. So maybe for if you choose blue and red or blue and violet, but eventually it have flow homes that can be excited by the laser, like Aviva said, and are unique, has a unique chain signature. They can be run together. So if you have, for example, 14 detectors, if you have 14 unique flow homes, you would be able to run them together. So I think if there are no further questions, we'll be happy to continue any discussion by email or on the phone. We are available, of course. And we would like to thank you for joining us today, and have a great rest of the day. Bye-bye.
Bye-bye.